What's up, everyone? How are you? How you doing? I hope you're doing well. Today we're talking about one of my favorite enzymes, and this is glycogen phosphorylase. Now, this enzyme is crucial, as we'll see, in metabolism. So we'll get to talk about it in details in the coming chapters when we get to talk about metabolism, like glycolysis and so on and so forth. But today I want to look at it as an enzyme, as an example of an enzyme. We've been looking at enzymes for weeks now, so you should know how an enzyme works, and we're going to look at it as an example of how it works, how it's turned on and off, that's regulation. And we're going to look at the structures and things in the structure that make it easier to control, if you will. So we're going to look at all of this and more. So make sure that you take good notes, make sure that you look at this video, and let's do it. Alright, so let's go back to glycogen phosphorylase. So if you look at this is an example of glycogen metabolism. A lot of people look at glycolysis and they think that glycolysis is the main key, but you have to remember that most of your blood and body glucose is stored as glycogen. And a lot of times, instead of breaking it down to glucose, we break it down to glucose 1 phosphate. Now, why would we do that? We would do that because glucose 1 phosphate is a molecule that's ready to be converted into energy. Glucose is not, and we'll talk more about it when we get to glycolysis. So the number one step in glycolysis usually is to convert glucose to glucose 6 phosphate. But if you have glucose 1 phosphate, it's much easier. So that's where glycogen phosphorylase is used. And as we'll talk later, you'll see how this glucose 1 phosphate is used in many different pathways. So you have the glycolytic pathway, you have the pentose phosphate pathway, and all these other pathways that we'll talk about in the future. But it all starts with glycogen phosphorylase releasing one glucose molecule from glycogen. How does it do that? We'll talk about it in a minute. Okay. So glycogen phosphorylate, and we'll call it GP as well, is an example of many enzymes that are regulated by allosteric control. So we've talked about allosteric enzymes. This is a big example of an allosteric enzyme, and it's regulated by both allosteric controls and covalent modification. So this is a good example for allosteric and covalent modification that we looked at. And its role, as we just say, it cleaves glucose units from non-reducing end of glycogen. So remember when you talked about glycogen and glucose, we talked about reducing and non-reducing end. So it removes it from non-reducing ends, and it converts glycogen, again glycogen, takes the glucose molecule and turns into a readily usable fuel in form of glucose 1 phosphate. And this is a phosphorylation reaction. So when we look at its structure, this is a dimer of identical subunits. So it's a homodimer, right? It's a homodimer and it has an allosteric site. So let's look a little bit at the mechanism where you have a long chain of glucose, right? Like we've seen, and then you look at the non-reducing end. Remember the non-reducing end. So we take the non-reducing end here, right? On this end, we're going to remove this group, right? We remove that group and then we add phosphate there. That is the reaction, right? And when you look at the reaction in detail, the phosphate here usually has a OH, so usually has 3 oxygen and another oxygen here. So this is POH, right? So this H group is going to attack this carbon. I can't see it very well. No, that's not the carbon I want to point at. Let's see if I can. I'll have to move it. Alright, so we attack that carbon. There you go. And then when we attack that carbon, it's going to break this bond right there. And when we break that bond, that will be ready to be attacked by phosphate. So it's a two-step mechanism, and that we end up having a product like this where you have glucose 1-phosphate, right? So this is the reaction, and whenever you have glucose 1-phosphate, by the way, as you've noticed here, it's not the one that is used. So you need to convert it to glucose 6-phosphate to interglycolysis and all these other pathways. So what we use phosphoglucomutase. So we remember that mutases are these isomerases, right? So the phosphate group moves from glucose 1 to glucose 6, right? So glucose 6-phosphate is this really important molecule, and that's how we make it. All right, so again, this is the importance of it. It's really important. We'll get back to how it's important, but again, today we're talking about its role as an enzyme. So let's get back to it. So again, it's a dimer. It's a dimer with identical subunits. Each subunit has 842 residues or subunits, right? And each one has 3 different sites, guys. 3 different sites. What are the 3 different sites? You have an active site, right? Then you have an allosteric effector site. That's number 2. And then you have a phosphorylation site. That's number 3. So you have 3 sites on each subunit, right? Which is pretty cool. And also you have another 4th site where you have a tower. So you have a tower at the end, and both subunits get together and they make what we call like a tower helix. So they make 2 little towers. The 2 towers, they get together and they do make 2 little towers, and those are the 4 structural features of the enzyme, right? So this is an example of it where it's showing you the allosteric site, the tower helix is here, you have the catalytic site here, and then you have the other site, that's the glycogen binding site where the glycogen binds. So that's an example of just one subunit. Actually, let me see if we can pull up the actual structure of it. Let me see. There you go. So this is a structure. We can move it around. So these are the 2 subunits right here, right? 2 subunits moving around, but it's hard to see. But again, by the way, you can use Chimera to move this around and look at different subunits. I'm looking, for example, at the 2 helices, 2 tower helices, right here. But again, another thing that you can do in Chimera is you can select. So I can select one chain, and then I can color it a different color. Let's call it purple. Right, so that's just one subunit, right? And then let's check the other one. Just want to see how it looks like. Let's color it green. Alright, so this can show you how the two subunits interface together. So this is one way to study it, but you can literally go in and study each subunit and each group and select them and see how they interface with each other. You can do all of that. You can see some of the amino acids we'll talk about, like some the amino acids that are phosphorylated are in there, are selected in here, right? These are the ones that are sticking out that you see in there. Some that are phosphorylated. You can see, I don't know if I can see it very well, but I can see inside that you have a substrate. I think we do have a substrate. Um, but you can use this to study this structure and see all of these structural features. I just wanted to show you that you can do that, but it takes time to play with it. You can I don't know if you can see my whole screen, but if you go to tools, go to sequence, you can actually select different sequences, right? Let's see if you can see. Oh, you can't see my screen. You can select different sequences, like specific sequences, and then you can color these and study them differently. So I can color this red, and then I can study it, right? So you can do so much in here to study different structural features. And this is Chimera. It's a free software that you can do anything with. And when you do this, you can make very, very beautiful images that you can use for publication. You can make movies. You can do all of this stuff um, just by using this software. But anyway, let's go back to glycogen phosphorylase. So 4 structural features that you can look at. Now, when you look at the kinetics of this enzyme, you'll see that, let me go back and remove this now. Yes. So when you look at the kinetics of this enzyme, you see that you have cooperativity. You have a cooperative binding. And where do we find cooperative binding? In allosteric enzymes. So this is weird binding compared to your normal Michaelis-Menten kinetics. This is the one that uses a sigmoidal shape. So the weird one that uses sigmoidal shape, this is when you have cooperativity, right? And that tells you that there is cooperativity between the two subunits. It tells you that there is allosteric regulation going on. Now look at this. ATP and glucose 6-phosphate are allosteric inhibitors of glycogen phosphorylase, and this is important. Why is it important? Because here, when you've made a lot of ATP and a lot of glucose 6-phosphate, you want to go back and inhibit glycogen phosphorylase. So that's what the enzyme does, right? So whenever you've made too much of this, it goes back and inhibits this, right? Or you've made too much of ATP, right? ATP is made here, by the way. ATP is made after, when you go through glycolysis, CO2 fresh H2O is plus ATP, right? ATP is something, that's what we make mainly. Okay, let me put plus ATP here. Plus ATP. So if you made ATP and you make too much of it, it goes back all the way up and it tells this enzyme like, hey, slow down, we're making too much. So these are the two main inhibitors of the enzyme. So these are allosteric inhibitors, right? And what is an activator of it? AMP is an activator. Why is AMP an activator? It's because AMP is a signal for low energy in the cell. It's a signal for low energy in the cell. So if you have high AMP in the cell, it shows you that you have low ATP and you need energy. And then what do? You go back and you tell glycogen phosphorylase to go and break down your fats so that we can start making energy, right? So AMP is a really good activator for this, right? So ATP, glucose, ATP, glucose 6 phosphate are inhibitors. Caffeine is an inhibitor too as well. So going back to cooperativity, remember that this is the typical sigmoidal shape of a typical allosteric enzyme. So this is an inhibited enzyme. Right? The second one is when you add ATP or glucose 6-phosphate. What happens? We inhibit it, right? And remember that it goes back to the right, right? And if we had to calculate the K of the enzyme, remember that it's going to be high. It's going to be higher, right? And whenever you inhibit the Km goes higher, and when the Km goes higher, what happens? The enzyme is less effective, right? The enzyme is less effective. And the opposite happens when you add AMP. When you add AMP, the enzyme is better, right? The enzyme is faster. It has a higher affinity for the substrate, right? By the way, the substrate here is the phosphate. So Pi is the phosphate, the free phosphate molecule. That's what they're using, right? All right, so that's how you regulate it. And let's talk about more regulation. So the active form, right? So it follows the AMWC model or the concerted model that we talked about, right? So the concerted model, remember, in the concerted model, the active form of the enzyme is the R state or the relaxed state. The inactive form of the enzyme is the T state or the taut state, right? So what does AMP does? AMP, which is an activator, promotes the conversion from T to R, right? And then the ATP, glucose, and caffeine, they favor the conversion from R to T, right? That's pretty much how the concerted model does, right? And there is a conformational change that happens, right? That happens between, the conversion between T and R, right? And we talked about that. But the main thing is the structural changes happen at the active site, right? They happen at the active site of the catalysis. So they happen at the active site. All right, so there is another layer of regulation, right? So let me put this up so that you can see it. There's another layer of regulation that happens. And what's the layer of regulation? Is covalent modification, right? Covalent modification is an important layer of modification when it comes to phosphorylase. Why? It's because you have two versions of phosphorylate. You have phosphorylase A, you have phosphorylase B, right? And phosphorylase B is unphosphorylated. Phosphorylase A is phosphorylated, right? And then that's covalent control. We can turn it from B into A, and these are structurally different, and these do different things. They do different things, they act differently. And then after that, at the bottom, you have what? You have allosteric control. So you have covalent control and then allosteric control, which is the non-covalent control. What's the allosteric control? Is when you add AMP, you activate it. When you add glucose 6-phosphate, glucose, caffeine, or ATP, you inactivate it. Right? So you have activators and inactivators. That's regulatory control. So remember, two types of control. You have covalent control and regulatory control, right? And this covalent control, let's talk about it a little bit more, all right? So again, this is an example of the phosphorylation site at the serine 14, right? So let's talk about this regulation a little bit. So in the 50s, that's where they found that there is an enzyme that converts phosphorylase B to phosphorylate A, right? And then later they found out that the conversion happens by phosphorylation. So the enzyme was named kinase. Phosphorylase kinase. And they found out that it's not just the phosphorylase kinase, by the way. So the phosphorylase kinase, which is a kinase, it comes in and phosphorylates our phosphorylase, which also does the phosphorylation. So, this is like a cascade, right? It is phosphorylated as well. But they found out that it's more than that. So it's even a bigger cascade. So it looks like this. So, so what does this mean? Our phosphorylase is here. It's converted from inactive to active, right? No, actually, it's not that. That's not our phosphorylase. Our phosphorylase is down here. That's the glycogen phosphorylase. That's the one we want to convert from B to A. That's our phosphorylase. But to convert it, we need phosphorylase kinase. Phosphorylase kinase converts it, right, from B to A. This is the phosphorylase kinase. And guess what? Phosphorylase kinase needs to be active as well. It needs to be active for it to be able to phosphorylate this. How is it activated? By phosphorylation, of course. So it also is activated by an enzyme, right, called cyclic AMP dependent protein kinase, right? So that is the one that also needs to be activated. How is it activated? By cyclic AMP. How is cyclic AMP activated? Cyclic AMP is the only one that's not an enzyme in this cascade. It's a molecule. It's not an enzyme, it's a messenger molecule. How do we get cyclic AMP? We get it from ATP. How does that happen? It happens through an enzyme called adenylyl cyclase. Adenylyl cyclase takes ATP and converts it into cyclic AMP. And cyclic AMP, by the way, it's a molecule, you've seen it before. I'll show it to you later, but it's just a AMP in a circular form. So adenylyl cyclase, how is it activated? It is activated from outside the cell by hormones and other things. So cyclic AMP again is you take ATP, right? You use the enzyme. In this case, it's called adenylyl cyclase, and you can see they attack by a base, right? Where we want the oxygen, the oxygen at the carbon 3 at the bottom is going to go and attack the phosphate so that we make a cycle. So cyclic AMP looks like this, right? So it's just AMP, right? It's adenosine monophosphate, but the phosphate binds to that oxygen at the bottom there. That's cyclic AMP, no problem. All right, so cyclic AMP is a really important molecule, and it's a messenger. It's a second messenger. What does that mean? It means that usually hormones and neurotransmitters are messengers in our body, but cyclic AMP, we call it a second messenger because it takes the message of those things and then it transmits into the body, because these things usually don't enter the body. They enter receptors at the bottom, they give their message, and one of the things that carry their message is cyclic AMP. It's really good at that, right? So it takes a lot of messages from hormones, and then the hormones like to bind to things we call GTP binding proteins or GTP binding receptors, and this is a whole nother area of uh, research that I could talk about for days. But remember this name, GCPR, G coupled protein receptors. These are really important receptors. And this is an example. This is an example of G-coupled protein receptors. So you have a hormone. Hormone binds to the receptor. You can see it, right? The hormone binds to the receptor, and then after the hormone binds to the receptor, it transmits the signal to these G proteins. The G proteins are, there's a whole family of G proteins. And they have three subunits here. They are activated and so on and so forth. They move their signal to adenylyl cyclase, and then adenylyl cyclase takes ATP, turns into the cyclic AMP, and then the cyclic AMP goes on to transmit the message, and then the rest of the cycle is this going back to the original form. And this is a common, very, very common mechanism that we see a lot. So this usually looks like this, right? This is your typical G protein. So this is an adrenaline receptor. So adrenaline is one of the big ones here, where adrenaline comes in, activates adrenaline receptor, it activates the G protein, adenylyl cyclase, and all that stuff. So this is a typical mechanism, and this is an example of your typical G protein coupled receptor. This is a family of a lot of proteins that are like that. A lot of drugs have been made targeting this mechanism. So hopefully we'll get a chance to look at this mechanism and talk about it because it's very interesting. But for now, we were talking about um, glycogen phosphorylase, and I hope that you understood what it's about, what it does, how it's controlled, how it's regulated, and you should be able to also know the mechanism, the structural features, and all of those things regarding glycogen phosphorylase. And if you have a question, you know how to reach me. I'll see you next time. Bye.